Neuroplasticity is kind of a two-edged sword. So it can be good and it can be really bad. So all it means is that there's a period of time early on in life where we have maximum ability to change our brains so that the environmental stimuli that come in have maximum effect. So that means that if all the right environments are in place, we can move the brain more so early on in life than later in life towards good outcomes. But the opposite is true too. So we know kids who are traumatized early on in life get stuck with adult phenotypes, adult disorders, because that window of neuroplasticity was so sensitive that it moved the shaping of their brain towards a worst outcome. But neuroplasticity is not only in early life. So that's your largest window. This is a time where you can have the maximum effect. And that's why we're talking about the earlier the diagnosis, the better the effects. But our brains are neuroplastic throughout our lifespan. And that's why we learn. If they were not plastic, we would not be going to school, right? We would not be learning anything if there was no plasticity. So the capacity to change the brain is there for our whole lifespan. It's just at its maximal for the first 4 or 5 years of life or so. There are certain drugs that change neuroplasticity, but again, we've been very careful with those because if we open the window of plasticity, we can do good, but we can also do a whole lot of harm. Because all it says is you can move the brain faster, but it doesn't protect you one way or another. Neuroplasticity is probably not as much related to making new neuron cells, and some of you asked me about that. We do make neuron cells for the rest of our life in certain parts of the brain, like the hippocampus, where is our memory and learning center. And we know for a fact that when we stop that, the effects are detrimental. So kids who get whole brain irradiation for brain tumors, for example, we partly kill the ability to generate new neurons in the hippocampus, and their IQ keeps dropping with age. So we know it looks like you need to be generating new neurons to be keeping your IQ up, but the majority of the neuroplasticity is not about making new neurons. The majority of it is wiring, as some of you have already highlighted. And definitely there is ability to rewire the brain. That's the basis, the biology of the learning process. And so there is no reason to believe that there comes a time in life where we don't have adequate kind of infrastructure in our brain to learn new things. We know people who retire, pick up new languages, pick up new skills. That's all based on neuroplasticity. So neuroplasticity is there for good, for the rest of your life, unless you are unfortunate enough to get significant amount of irradiation into your brain. But it is true that the earlier we intervene, the better, because the window for plasticity is bigger earlier on in life. There are all kinds of ways we can take advantage of plasticity, and that's where most of the training and intervention curricula are based on. So in the case of ASD, the whole early intervention system is based on the idea that we have neuroplasticity. In the ADHD world, you're going to hear about brain training. The whole idea is that there is still neuroplasticity, and so on. Any learning, for it to take place, we have to accept that there is neuroplasticity, otherwise we wouldn't be able to learn. Environment and genes. Okay, tons of questions on environment and genes. I don't have a satisfying answer, but this is my answer. So all of these disorders have genetic predispositions, for sure. Some of them even a bit stronger than others, but all of them have a very significant genetic component. When we say genetics, we don't necessarily mean heritable. So genetics doesn't mean that you got it from your parents. Genetics means that you have a difference in your genome that puts you at risk, right? So in the case of autism, only 50% of the kids who have genetic mutations actually got it from a parent. The rest of the kids, it was a random error that happened at the time of conception. But genetic means that you start with a genetic difference that, as Nii described in the morning, will likely lead to a change in your proteins, which are the workers in the body, so you're going to end up with a different brain, a different body. Most of these things don't matter. Blue eyes, brown eyes, tall, short, doesn't matter, right? Some of those matter because they produce dysfunction. And that's the only time when we consider a genetic difference pathogenic or related to disease, when it produces dysfunction. So then you get the other balls into the bucket that Nee showed you, right? So you have your genetic predisposition, and now you have to figure out where the environment, which is clearly affecting your brain, right? Because it's part of what contributes to neuroplasticity, the pathway for neuroplasticity, has to interact with these genes to produce what it's going to ultimately produce. Now, an environment to interact with the body. It can do it a couple of ways, but the most common way that it happens in neurodevelopmental disorders is it talks through genes. So we have a pathway that's determined by genes, and then we get an environmental influence on that pathway. That's why in most of the neurodevelopmental disorders, and there are some exceptions to the rule, we talk about mostly gene by environment interactions. It's not like we walk out on the street, we get exposed to some telephone pole or whatever people are worried about, and all of a sudden the baby has autism. For environmental toxicant, even when it's a good one, and there are lots of bad candidates out there, you need some kind of biological pathway that's likely determined by genetics that it will interact with. And that's why we don't have a single environmental agent yet that can produce autism on its own. Like, we have no evidence of a single toxicant that is adequate to produce disease. There are autism or a neurodevelopmental disorder. There are catastrophic events, right? So there was a huge talking about mercury spill in Iraq many, many years ago from a factory. The kids died there. They were not having autism or ADHD or developmental disorders. They got strokes and they died, right? So there is a possibility to have catastrophic environmental events. But for the most chronic interactions that we're talking about environment by gene interactions. For those of you who ask me for specific examples, I'm going to be very nervous, but I'll bring you a couple. So again, if you think about environment, you cannot think only after birth. The most important environment the brain gets exposed to is the fetal environment, right? So baby in mom's womb is the environment. And so many, many things can happen during pregnancy that would produce an environment by gene interaction that may not be favorable. Things like, we can have a flu epidemic. During flu epidemics, we have a slight increase in neurodevelopmental disorders. During, if mom is exposed to a specific drug that seems to be changing brains towards the direction we don't like it, we do get a spike for autism. So for example, valproic acid, although we use it in kids quite a bit, if the fetus is exposed during pregnancy to valproic acid, the babies have a higher chance of having a neurodevelopmental disorder. There is some early data to suggest that gestational diabetes and other metabolic complications during pregnancy may slightly affect the risk. Now, the actual effect on each child is very small. That's why we're talking about a gene by environment interaction. You have to have the genetic predisposition. But in the population, moms who have gestational diabetes have a higher incidence of having babies who have neurodevelopmental disorders than those who don't. There are several metabolic pathways that are candidate pathways that people are studying, but we know that this is happening, right? And then after birth, somebody asked me about perinatal injury, so acute birth injury, and I would say the evidence we have so far is not strong at all. So when you get a severe acute injury during birth, you either kind of make it or don't. So either kids get a severe encephalopathy and they end up with strokes and CP and so on, or they have good outcomes. But the prolonged complications of the third trimester have been proposed to actually affect neurodevelopmental outcomes. So for babies who have an abnormal fetal circulation, let's say, and affects brain development during the third trimester, if they have a congenital heart lesion or something like that, that will increase their chances of getting autism spectrum disorder, ADHD, intellectual disability. So there are a few factors that we know around the time of pregnancy, and some after pregnancy, although for those we're not as convinced, that we know if they interact with the wrong combination of genes, will increase the chance for somebody to develop a neurodevelopmental disorder. We have to study this. The approach that we are taking for POND is that instead of just looking at all the environmental factors, we start from the genes that have paths that we know are amenable to environmental manipulation, and we study them one by one. So it's tedious and it's slow, but at least at the end of it, we're hoping you can actually believe what we show you. I'll take maybe another couple. The Duke study. So somebody brought up the Duke study. So I want to make a point about that study. So it's not about the Duke study. It's about this whole business of autologous cord blood transfusions. They're not specific to autism. People with ADHD get them. Babies with other neurodevelopmental disorders are going to Mexico these days to get cord blood transfusions. So I just want to clarify something. First of all, it's not a gene treatment. So if you were wondering about this gene, it has nothing to do with gene therapy. The basic premise for why it may be worth studying something like that is that in people who have, mice and people who have a cord lesion, so they have disconnected fibers in the cord, when they get autologous cord transfusion, what they get is some growth of the myelin, which is the white matter that wraps around the nerves to help with connectivity. So somebody had the idea, and there is some other effects of those transfusions. They affect inflammatory markers, and we're going to talk tons about inflammation later. So somebody had the idea of whether if we were to give their cord blood, their banked cord blood, whether we would improve connectivity or manipulate inflammatory factors in a favorable way. It remains to be seen, but the experiment is being done, so we have to wait for the study to be done for us to have a sense of whether these types of approaches would be at all useful in our disorders. Because we don't have a cord lesion, right? We don't have a break in the connectivity. I have 2 minutes to go. So we talk about science mostly today. You're going to hear from our parent advisory group later, but we are invested in obviously affecting policymakers and the way we deliver healthcare systems, healthcare to our children. And so OBI has a whole agenda that's related to policy. So much above our heads, much above the head of pond, the Ontario Brain Institute is communicating with the ministries and has a whole group of people who are working on knowledge translation and policy, affecting policy. From the pond point of view, most of us are sitting on both provincial and federal committees. I can tell you it's very satisfying work, very slow, and you have to kind of put up with doing a lot of work for very little output, but we know that it's very important. And so most of us, you can come and talk to us after, we'll tell you exactly what we're working on, but are sitting on both provincial and federal committees for the disorders we have expertise in. And then our KT core, so for now, most of the work is on communicating to our public, so with the work that was presented, but the ultimate goal, obviously, is that we communicate to both policymakers and our families. And maybe I'll take just a DSM-5 question, because I'm done, apparently. So, well, actually two, very quickly. One is an obesity question, which is a very interesting question. So lots of kids across our disorders have obesity. And I got the question about whether this is genetic or environmental. And like everything else, I'm going to tell you it's a gene by environment interaction. So we have some genetic mutations that are shared among the disorders that are actually causing obesity. And it's a genetically predetermined obesity, and we have to work with the families to keep these kids healthy. But we also, in certain of the disorders, in some more than others, so probably more than in ASD than in ADHD, we have kind of the socially isolated, passive little boy, as a proper example, who doesn't participate in sports, who is low tone and is clumsy. So it's not easy for them to be active. So there is probably some type of gene by environment interaction, but there are definitely genetic predispositions to obesity in our kids. So my last comment about DSM-5. So you're going to hear divergent views on the DSM-5. I'm not sure if you pick all the investigators in POND whether we're going to agree. In fact, I know we're not. So you're going to get my bias here. So we absolutely, clearly this is heterogeneous. You saw in Jason's data, big brains, small brains, all under the same phenotype, mice and humans. Very heterogeneous disorder, right? We clearly understand the need to break it down to the meaningful subgroups, to study them and to develop treatments for them. Whether DSM-4 was the right way to do this is the question. And my bias is that it was not. So from what we understood from the biological studies we had, we could not make distinctions between the subcategories allowed by DSM-4. And so DSM-5 is almost an admission that we don't know how to do this. It's not that we believe this is a homogeneous disorder, but we're saying splitting it by, in the case of autism, PDD, NOS, Asperger, and autism, does not identify biologically distinct subgroups. It gives us the illusion that we know something we don't know. So let's all admit we don't know how to do this. We call it a spectrum and start from scratch studying biology. So I think it's going to affect funding. That's a reasonable point that James brought up, but I don't think the previous classifications helped us much. So it may be good to start from an agnostic point and say we just don't know. Let's figure out what the biologically meaningful groups are.